Assalamualaikum. So our group, subgroup 10, present about BLAST, and I will start with the introduction part. For the introduction, Basic Local Alignment Search Tool or BLAST is a program that able to identify similarity between a query sequence and sequences available in database. These programs allow us to detect supposed genes in a novel sequence and to figure out the relation of the proposed gene or protein to other non-genes or protein. It also widely used because it can instantly identify regions of local similarity between two sequences, and BLAST also uses a robust statistical framework that able to find out whether the alignment between two sequences are statistically significant. Next, I will pass to Nur Hazwani.
Now I will explain about the demonstration of how BLAST can be used in molecular biology or biotechnology. There are 4 steps. The first one is use BLAST in tools from the data and software in NCBI. Second, insert the nucleotide sequence into the query sequence box. The third one is choose the standard databases under the choose search set before click BLAST button to start analyze. And the last one is observe and analyze the identity of the sequence. So the first step, you go to the NCBI page as stated in the slide. And then you click the data and software from the list menu on the side of the NCBI homepage. After that, in the data and software side, choose the tools, and in the tools side, choose the basic local alignment search tool, which is BLAST. For the second step, insert the nucleotide sequence into the query sequence box. In the BLAST homepage, choose the nucleotide BLAST and then copy your sequence as stated in the slides, for example, and then paste the sequence into the query box form of BLATN, BLATN. For the third step, choose the standard databases under the choose search set before click the BLAST button to start the analysis. Where under the choose search set, choose the standard databases and etc. Then click the BLAST button. It will take some time for your results to be displayed. And for the last step, observe and analyze the identity of the sequence used. Based on the result, the nucleotide sequence from the example is predicted from the Oriza sativa. Next, I will pass to Sharifah.
Okay, next let's proceed to BLAST application. In molecular biology, BLAST aid in drug discovery and identification of macromolecules identity, whereas in modern biotechnology uses BLAST to screen sequences for genetic engineering and discover new genes. Next. In drug discovery, BLAST detects biosynthetic pathway, functional and chemical interactions. Sequence comparisons provide a cascade to identify organism yielding final product or chemical pathway of medicinal interest. To identify macromolecules such as proteins and nucleotides, BLAST compare the unknown sequences with the one of similar sequences, as similar sequences subjected to similar function. In screening of nucleotide sequences for gene of interest, when sequence of known gene inserted as query, the results provide other source in which the genes can be extracted out for the purpose of genetic engineering of novel products. In terms of discovery of new genes, through NGS, transcriptomic and proteomics, query sequences of functional genes with low BLAST results indicative as new genes that may arise from natural mutation or genetic engineering. Next, I will pass to Nisa.
Moving on to the comparison of its usefulness to other similar software. Next. Here I will be comparing BLAST to another software called BLAT. BLAST, or else Basic Local Alignment Search Tool, and BLAT is a BLAST-like alignment tool. BLAST builds an index of query sequences and searches through the database for matches. It also allows lower scoring hits and gives access to more gaps in alignments, though it may be slower. whereas blood, it indexes the genome or protein database. It retains the index in memory and then scans the query sequences for matches. It is fast, but it requires more accurate matches. Next.
Next, I will be comparing the next comparison, where BLAST accept both FASTA and queries by accession number. Meanwhile, BLOOD requires query sequences in FASTA format. BLAST can find much more remote matches than BLOOD, and it can be used to infer functional evolutionary relationships between sequences and also to add in identifying members of gene families. Meanwhile, BLOOD, it needs an exact or nearly exact match to find hit, and therefore, BLOOD is not as flexible as BLAST. BLOOD is also not as sensitive and not as accurate as the tool for analyzing sequence alignments. So in conclusion, BLAST has become an essential tool for biologists or biotechnologists. Its speed and sensitivity allows scientists to compare nucleotide and protein sequences to both single sequences and large database. And I think that's all from us. Thank you.